Welcome to the Deep Dive, where we take complex topics and, well, basically give you the essential insights and surprising facts you need to be truly well-informed, fast. Today, we're plunging into the fascinating world of carbohydrates, and we're starting with something you might literally have in your medicine cabinet right now.
That's right. Did you ever stop to wonder what neosporin actually is? You know how it works?
The antibiotic cream.
Exactly. And one of its key active ingredients, neomycin, is fundamentally a derivative of a carbohydrate.
Wow, neomycin, a
Mhm.
carbohydrate derivative. It's quite a twist for, an everyday item. And it perfectly sets the stage for our deep dive today. Our mission here is to explore these molecules that are just everywhere, from their basic building blocks and how we classify them,
Right.
through their sometimes surprising reactions, all the way to their absolutely crucial roles in nature. Being the very foundation of life itself.
Absolutely fundamental.
So you'll uncover key concepts, you'll see how these molecules transform, and their really profound real-world applications. Let's get into it.
Okay. So when most people hear carbohydrates, like you said, they think food, pasta, bread, sugar.
Yeah, the energy stuff.
Right. But chemically speaking, what are these compounds?
That's a good question. We use the term constantly, but what's the actual science?
Well, at their most fundamental level, carbohydrates are polyhydroxyaldehydes or ketones.
Okay, polyhydroxy. Lots of OH groups.
Exactly. Multiple hydroxyl groups, those OH groups, plus either an aldehyde or a ketone functional group. And interestingly, the name carbohydrate is, it's a bit of a historical leftover.
Oh,
Yeah, for a long time, chemists thought they were basically hydrates of carbon. If you look at glucose, for example, its formula is C6H12O6.
right.
You can rearrange that to look like C6H2O6, 6 carbons, 6 waters.
Ah, I see. Like carbon plus water.
Precisely. We know now that's not how they're actually structured, but the name stuck.
So, a name based on an old misunderstanding. But what's really incredible is just how abundant and, well, essential these molecules are.
Oh, absolutely. Think about it. A huge chunk of the food we eat is carbohydrate. It fuels almost all our biochemical processes.
Right, the energy source.
Glucose is the classic example. Plants use photosynthesis, right? They take carbon dioxide and water, use sunlight energy, and bam, they make glucose. They're essentially storing solar energy in those chemical bonds.
Capturing sunlight in the molecule.
Exactly. Then, when we eat plants or things made from plants, our bodies break down that glucose, convert it back into CO2 and water, and release that stored energy to power everything we do.
It's such an elegant cycle. Solar energy captured, then released inside us. But it's not just about energy, is it? Carbohydrates are structural too.
Precisely. They're critical for rigidity in countless organisms. The wood in trees, that's mostly carbohydrate.
No.
The hard shells of lobsters, crabs, also carbohydrate-based. Chitin, specifically.
Wow.
And here's where it gets really fundamental. Even the building blocks of our DNA and RNA are genetic material. They're derivatives of carbohydrates.
From a tree trunk to your actual DNA. That's an incredible range. So how do chemists even start to classify these things? We hear about simple sugars all the time.
Right. Simple sugars are called monosaccharides. That literally means single sugars. The word comes from saccharum, which is Latin for sugar.
Makes sense.
And when these monosaccharide units link up, they form more complex sugars, things like disaccharides with 2 units or polysaccharides with many, many units. We'll definitely get to those later.
Okay. And within those monosaccharides, those single units, there's a distinction between aldoses and ketoses, right?
That's right. You can usually tell it's a carbohydrate by the -ose suffix at the end of the name, like glucose, fructose.
Got it.
The difference comes down to a key functional group. An aldose has an aldehyde group. Glucose is the prime example there.
Aldehyde, aldose. Okay.
And a ketose has a ketone group. Fructose is the classic example of a ketose.
Ketose. Simple enough.
And then we classify them further by the number of carbon atoms they contain. We use prefixes like tri for 3, pent for 5, hex for 6, right before the ooze.
So you could have like an aldotose,
Yeah,
an aldehyde with 5 carbons.
exactly. Or ketohexose, a ketone with 6 carbons like fructose. It helps us be really specific.
Okay, that helps decode those complex names. Now, something that always tripped me up in chemistry was the D and L designation.
Yeah.
It seems confusing.
Yeah, it's a really common sticking point. The system we use, the Fischer-Rosenoff convention, it uses glyceraldehyde as the reference. That's the smallest carbohydrate with a chiral center, a handedness.
Okay.
The crucial thing to understand is this. The D designation means that when you draw the sugar in a specific flat way, called a Fisher projection, the hydroxyl group, the OH on the chiral carbon farthest from the carbonyl group, points to the right.
Farthest from the top group, pointing right.
Okay, it's basically defining a standard handedness for families of related sugars. Most naturally occurring sugars are D sugars.
But, and this is the tricky part, I remember, D doesn't actually tell you which way it rotates light, does it?
Exactly. That's the big aha moment. It's super important. For example, D-erythros and D-threos are both D sugars by structure, but they actually rotate plain polarized light to the left. They're liber-rotatory.
Oh, wow.
Okay. So D and L only tell you the configuration, the 3D arrangement of that specific chiral center relative to glyceraldehyde. It has nothing to do with plus or optical rotation. That's a separate experimental measurement.
That's a really vital distinction to make. So keeping that system in mind, what are some of the, the star players among simple sugars?
Well, among the D-aldehexases, the 6-carbon aldehyde sugars, D-glucose is definitely the most common and arguably the most important.
The king of sugars.
Pretty much. And among the D-ketohexases, D-fructose, the main sugar in fruit, is the most common one found in nature. And we absolutely have to mention D-ribose.
Ribose.
Yeah.
That sounds familiar.
It should. It's a 5-carbon aldose and aldopentose, and it's a absolutely key building block of RNA. And a close relative is deoxyribose, the sugar in DNA.
Right. The backbone of genetics. Okay, so we've defined them, classified them, but how do they actually exist? I remember hearing they're not always just straight chains.
That's exactly right. That open chain form we often draw first, it's actually a minor component in solution. Monosaccharides mostly exist as cyclic forms, as rings.
They curl up on themselves?
Yeah, basically. It happens through an internal reaction. A hydroxyl group within the molecule attacks its own aldehyde or ketone group. This forms what we call a hemiacetal, or a hemiketal link, closing the chain into a ring.
And is there a preferred ring size, like white rings?
Stability. The most common and stable rings are 6-membered ones, which we call pyranoses, named after a simple 6-membered ring compound called pyranate.
Pyranoses. Okay.
And 5-membered rings called furanoses, named after furan. These sizes are favored because they're relatively strain-free. Think back to cyclohexane rings, how they adopt those chair conformations to minimize strain. Same idea here.
Okay, strain relief. And when a sugar forms this ring, doesn't something new happen at the carbon that was the aldehyde or ketone?
Yes, absolutely. A new chiral center is created right there. This specific carbon is called the anomeric carbon. For aldoses, it's usually carbon number 1.
Anomeric carbon. And this new center can be arranged in 2 different ways.
Exactly. The 2 different stereoisomers that result differing only at this anomeric position are called anomers. We label them alpha and beta.
Okay, alpha and beta. How do we tell them apart?
It's about the position of the new hydroxyl group on that anomeric carbon relative to another group on the ring, specifically the CH2OH group that sticks off the ring for aldehexases. In the alpha anomer, that anomeric OH is trans to the CH2OH group. Think opposite sides of the ring if you imagine it flattened.
Alpha is trans. Got it.
In the beta anomer, the anomeric OH is cis to the CH2OH group. Same side.
Beta is cis. Okay. And these anomers, they don't just pick one form and stick with it, do they?
No, they don't. They constantly interconvert in solution. This process is called mutarotation.
Mutarotation. Sounds dynamic.
It is. The ring momentarily opens back up to that transient open chain aldehyde or ketone form, and then it can reclose as either the alpha or the beta anomer. It's like this little equilibrium dance happening all the time.
And this changes the overall optical rotation of the sugar solution.
Precisely. As the proportions of alpha and beta shift towards equilibrium, the measured rotation changes. And this whole process is sped up by acid or base.
So let's take D-glucose again, our most common sugar. How does this play out?
Glucose is a fantastic example. In solution, it turns out that the D-glucopyranose form predominates. It makes up about 63% of the mixture at equilibrium.
63% beta. Why so much?
This is a really key insight into why glucose is so special. In its most stable chair conformation, the beta anomer of D-glucopyranose can arrange itself so that all of its bulky substituents, all the OH groups and the CH2OH group, are in equatorial positions.
All equatorial, like sticking out to the sides, not bumping into each other.
Exactly. Minimum steric strain. It's the most stable possible arrangement for a 6-carbon sugar. This inherent stability is a big reason why glucose is so ubiquitous and important in nature.
That makes perfect sense. Maximum stability. What about fructose? Does it do the same thing?
Fructose is interesting. It can form both pyranose, six-membered, and furanose, five-membered rings. In solution, the pyranose forms tend to dominate, but the beta-furanose form, that five-membered ring, is actually really important in many biochemical pathways inside our cells.
So the form that's most stable in a beaker isn't always the one used biologically.
That's often the case, yeah. Biology selects for what works in its specific enzyme pockets and pathways.
Okay, so these sugars are dynamic, forming rings, interconverting. Let's talk about how chemists can intentionally modify them. How do they react? What if you want to change those hydroxyl groups?
Right. Well, one challenge with carbohydrates is that they're usually very soluble in water, which can make them hard to purify. A common strategy is to convert all those hydroxyl groups into esters or ethers.
Why would you do that?
It changes their properties, particularly their solubility. Ester or ether derivatives are often much less soluble in water and more soluble in organic solvents. This makes them way easier to isolate, purify, and handle in the lab. It's like giving them a temporary chemical disguise.
Clever. Changing their coat to pull them out of the mix. Now, what about forming something called a glycoside? That sounds important.
Extremely important. Glycosides are formed when the cyclic hemiacetal form of a monosaccharide reacts with an alcohol in the presence of an acid catalyst.
Okay, sugar plus alcohol plus acid.
And the key thing is that only the hydroxyl group on the anomeric carbon gets replaced by the alcohol group, forming an OR group. The other OH groups stay put.
Only the anomeric one. Why just that one?
The mechanism involves the anomeric OH group getting protonated and leaving as water. This creates a resonance-stabilized carbocation intermediate, but stabilization like that is really only effective at the anomeric position because of the adjacent ring oxygen. Then the alcohol attacks that position.
Okay, so it's chemically favored there. What's the big deal about glycosides then? Why are they so important?
Stability. Unlike the original monosaccharide, which is constantly undergoing mutarotation, opening and closing,
Right.
a glycoside is locked in its cyclic form. It's an acetyl, not a hemiacetal. Under neutral or basic conditions, that ring won't open. It does not undergo mutarotation.
Locked shut, so there's stable building blocks.
Exactly. This stability is crucial. It allows sugars to be linked together reliably to form disaccharides and polysaccharides, or to be attached to other molecules like proteins or lipids without constantly falling apart or changing form.
Okay, stability is key. Now, you mentioned basic conditions. What happens if you expose carbohydrates to strong base? You hinted earlier that might be problematic.
It definitely can be. Under strongly basic conditions, things can get messy. A base can actually pull off a proton near the carbonyl group in the small amount of open chain form that's present. This leads to an intermediate called an anediol.
Medial.
And this anediol intermediate can actually reprotonate differently, leading to the formation of other sugars. For example, D-glucose can interconvert with D-mannose under strong base. They're epimers.
Epimers. What are those?
Epimers are diastereomers, stereoisomers that are not mirror images that differ in configuration at only one chiral center. Glucose and mannose differ only at carbon number 2.
So you start with glucose and end up with a mix of glucose and mannose.
And potentially other sugars too, like fructose. It's why chemists generally try to avoid exposing carbohydrates to strongly basic conditions if they want to keep their sugar pure.
Good practical tip. Avoid strong base. What about redox reactions? Can we reduce or oxidize these sugars?
Oh yes, you can reduce the carbonyl group, the aldehyde or ketone. A common region is sodium borohydride, NABH4. This converts aldoses and ketoses into alditols, which are polyhydroxy alcohols.
But wait, if most of the sugar is in the ring form, how does the reducing agent get to the carbonyl?
Le Chatelier's principle in action. There's always that small amount of open chain form in equilibrium. As the NABH4 reduces that tiny fraction, the equilibrium shifts to open more rings to replace it. Eventually, the whole sample gets reduced.
Sneaky. So it just keeps opening rings until it's all reduced.
Yeah.
Any real-world examples of alditol?
Sure. D-glucitol, which you get from reducing D-glucose, is commonly known as sorbitol. You find it used as a sugar substitute in lots of diet foods and chewing gum.
Sorbitol. Okay, so that's reduction. What about oxidation?
Oxidation can happen at a couple of places. If you use a mild oxidizing agent, like bromine water, buffered at around pH 6, you can selectively oxidize the aldehyde group of an aldose to a carboxylic acid. This forms an aldonic acid.
Mild conditions, just the aldehyde gets hit.
Right. And importantly, these conditions don't oxidize ketoses. So this reaction provides a nice, simple chemical test to distinguish between an aldose and a ketose.
Useful. What if you use something stronger?
If you bring out the bigger guns, like hot nitric acid, you oxidize both the aldehyde group at the top and the primary alcohol group at the bottom of the chain, if it's an aldehexose, for example. This produces an alderic acid, which is a dicarboxylic acid.
Oxidizes both ends.
Okay.
And this whole oxidation idea connects to something we hear about in food and health, reducing sugars, right?
Exactly. A reducing sugar is simply any carbohydrate that can act as a reducing agent, meaning it can reduce an oxidizing agent. Historically, tests like Toland's reagent, which gives a silver mirror, or Feeling's or Benedict's reagent, which give a reddish copper precipitate, were used.
So what makes a sugar reducing?
It needs to have a hemiacetal group that can open up to form a free aldehyde, or a hemaketal that can isomerize to an aldehyde. So basically, all monosaccharides, both aldoses and ketoses, are reducing sugars because they exist in equilibrium with their open chain forms.
But what about the glycosides we talked about earlier? The locked rings.
Good point. Glycosides are not reducing sugars. Because that anomeric carbon is locked in the acetyl linkage, the ring can't open under the mild basic conditions of those tests. So no free aldehyde forms and no reaction occurs. That's a key distinction.
Reducing means the ring can open. Non-reducing means it's locked.
Got it.
Okay, shifting gears a bit. Can chemists actually perform, like molecular surgery, can they lengthen or shorten the carbon chain of a sugar?
Amazingly, yes. There are classic methods for doing just that. The Kiliani-Fisher synthesis is a way to lengthen the carbon chain of an aldose by exactly one carbon atom.
Add one carbon. How does that work, basically?
It involves adding cyanide to the aldehyde group, then hydrolyzing the resulting cyanohydrin to a carboxylic acid, which is then reduced. The tricky part is that when you add the cyanide, you create a new chiral center.
See, two possibilities.
Exactly. You typically get a mixture of two epimers, two sugars that differ only at that newly formed chiral center, C2. For example, starting with the 5-carbon sugar D-arabinose, the Kiliani-Fisher synthesis gives you both D-glucose and D-mannose, the two 6-carbon sugars that differ at C2.
Fascinating. You build up complexity. What about going the other way, shortening the chain?
The classic method for that is the Vol degradation. It effectively removes carbon number 1 from an aldose, shortening the chain by 1 carbon. Unlike Kiliani-Fisher, it usually gives just one product.
And was this useful?
Historically, extremely useful. These chain lengthening and shortening reactions were crucial tools for chemists back in the day to figure out the exact structures and configurations of all the different monosaccharides. It was like solving incredibly complex molecular puzzles.
Incredible ingenuity.
Yeah.
Okay, so we've covered single sugars, their rings, their reactions. Let's build them up now. You mentioned disaccharides earlier. 2 sugars joined together.
Right. Disaccharides are 2 monosaccharide units joined by that stable glycosidic linkage we talked about.
The locked acetyl link.
Exactly. A common example is maltose, which is found in malt used in brewing. It's made of 2 ID glucopyranose units linked between carbon 1 of the first glucose and carbon 4 of the second. We call that an IO4 linkage.
2 glucoses, alpha link. Is it a reducing sugar?
Yes, it is. Because the second glucose unit still has a free hemiacetal group at its anomeric carbon, C1, that ring can open and close. So maltose undergoes mutarotation and is a reducing sugar.
Okay. Who has cellobiose? Sounds similar.
Cellobiose is also 2 D-glucose units linked 1,4, but this time it's a beta-1R4 linkage. It's actually the repeating unit you get if you partially break down cellulose. And just like maltose, because it has a free hemiacetal, it's also a reducing sugar and undergoes mutarotation.
Alpha link in maltose, beta link in cellobiose. Got it. And then there's lactose, milk sugar. That's 2 different sugars, isn't it?
That's right. Lactose is composed of a galactose unit joined by a 1R4 linkage to a glucose unit. Galactose is an epimer of glucose, differing only at C4.
Galactose plus glucose, beta link. Reducing sugar, too.
Yep. Same logic. The glucose unit still has a free hemiacetal, so lactose mutarotates, and is a reducing sugar. And this brings us, of course, to a very common real-world issue: lactose intolerance.
Yes. A lot of people experience that. What's the biochemistry behind it?
It's due to having insufficient levels of an enzyme called lactase in the small intestine. Lactase is what normally breaks down lactose into galactose and glucose so they can be absorbed.
So if you don't have enough lactase?
The lactose passes undigested into the large intestine, where gut bacteria have a field day with it. They ferment the lactose, producing gases like hydrogen, carbon dioxide, and methane, plus lactic acid and other irritants. This leads to the uncomfortable symptoms: bloating, cramps, diarrhea, gas.
That makes sense. And it's surprisingly common, right?
It really is.
Oh, yeah.
Globally, estimates suggest maybe 75% of adults worldwide have some degree of lactase deficiency. It's particularly common in populations of Asian, African, and Native American descent. In the U.S., it affects somewhere between 30 and 50 million people.
Wow, that's a huge number. So people manage it with diet.
Yes, either by avoiding dairy products or by taking supplemental lactase enzyme tablets when they do consume dairy.
Okay. Now, what about the sugar we all know best? Table sugar, sucrose. How does that fit in?
Sucrose is the odd one out among these common disaccharides. It's made of a glucose unit and a fructose unit.
Glucose and fructose. What's the link?
This is the crucial part. They are linked between the anomeric carbon of glucose, C1 in its alpha configuration, and the anomeric carbon of fructose, C2 in its beta configuration. It's an ellipth 1 or 2 linkage. Both anomeric carbons are tied up in the glycolytic bond.
Both. So neither unit has a free hemiacetal.
Exactly. And because neither ring can open up, sucrose is not a reducing sugar and does not undergo mutarotation.
That's a key difference, locked shut on both ends.
A very important distinction.
And this chemistry also relates to artificial sweeteners, doesn't it?
It does. Many artificial sweeteners are designed to taste sweet but avoid being metabolized like sugar. Take sucralose, which is sold as Splenda. It's actually made from sucrose, but 3 of the hydroxyl groups have been replaced with chlorine atoms.
Chlorine instead of OH.
Yep, and those chlorine atoms change its shape and chemical properties just enough that our digestive enzymes don't recognize it, and it passes through undigested. So sweet taste, 0 calories. Stevia is another example. It contains molecules where sugars are attached to a non-carbohydrate backbone, and again, we don't metabolize them.
Fascinating molecular mimicry.
Yeah.
Hey, beyond pairs of sugars, we get to the really big ones, polysaccharides.
Right. Polysaccharides are huge polymers, sometimes thousands or even millions of monosaccharide units linked together by glycosidic bonds. Cellulose is a classic example.
The stuff in wood and plants.
Exactly. It's made of thousands of D-glucose units linked by those Bowen-War glycosidic bonds, just like in cellobiose. These long chains line up parallel to each other and form extensive hydrogen bonds between chains. This creates incredibly strong, rigid fibers. That's what gives plants their structural integrity.
Beta links, and that's why we can't digest wood or grass, right?
Precisely. Right. We humans lack the enzymes, the glycosidases, needed to break those Bowen-War linkages. Cows and termites don't have them either, actually. But they have symbiotic microorganisms in their guts that do produce the necessary enzymes called cellulases.
They outsource the digestion. So what can we digest easily? Potatoes, rice, bread. That's starch.
Exactly. Starch is the main energy storage polysaccharide in plants, and it's also made of glucose units, but this time they're linked by alpha-1 more glycosidic bond.
Alpha links, that's the only difference.
That's the main difference in the primary linkage. Starch actually has 2 components. Amylose is a mostly linear chain of ON104 linked glucose. Amylopectin is similar, but it's branched. It has ON1 linkages branching off the main ON104 chain every 25 units or so.
Branched versus linear.
Yeah.
And we can break those alpha links.
Yes. Our digestive enzymes, like amylase in our saliva and pancreas, are specifically designed to hydrolyze those ON104 linkages very efficiently, breaking starch down into glucose that we can absorb and use for energy. The subtle difference between alpha and beta linkages makes all the difference in digestibility for us.
Amazing how one little stereochemical difference has such huge biological consequences. What about our own energy storage? We don't store starch, do we?
No, our equivalent is glycogen. It's structurally very similar to amylopectin, a branched polymer of glucose with ON104 and ON106 linkages, but glycogen is even more highly branched, with branches occurring maybe every 10 glucose units.
More branches. Why?
The high degree of branching means there are many more ends to the molecule. This allows for very rapid release of glucose units when our body needs quick energy, like during exercise. We store glycogen primarily in our liver and muscles.
Quick access energy reserves.
Yeah.
Makes sense. So carbohydrates, shape trees, fuel our bodies. But they're also deeply involved in health and even our genetics. Let's talk about amino sugars.
Right. Amino sugars are basically carbohydrate derivatives where one hydroxyl group, usually on carbon 2, is replaced by an amino group. NH2. A really important one is BD-glucosamine.
Glucosamine, I've heard of that supplement.
You have. It's a component of cartilage. But its N-acetyl derivative, where the amino group has an acetyl group attached, is the repeating monomer unit in keton.
Keton. You mentioned that earlier with lobsters.
That's the one. It forms the hard exoskeletons of insects, crustaceans, spiders, and also the cell walls of fungi. The amide linkages in N-acetylglucosamine allow for even stronger hydrogen bonding than in cellulose, making keton incredibly tough and resilient.
Stronger than cellulose. Wow. And this path leads us eventually to our own genetic code through something called N-glycosides.
Yes. Remember how a regular glycoside forms between the anomeric carbon and an alcohol's oxygen? Well, if the anomeric carbon reacts with an arcMon's nitrogen instead, you form an N-glycoside.
Sugar linked to a nitrogen atom.
Exactly. And two sugars form especially important N-glycosides, D-ribose for RNA and 2-deoxy-D-ribose for DNA. They link up with specific nitrogen-containing bases, adenine, guanine, cytosine, thymine, or uracil.
And that combination is a nucleoside.
Precisely. A sugar plus a base is a nucleoside. Add one or more phosphate groups to the sugar part of a nucleoside, and you get a nucleotide.
Sugar plus base, nucleoside. Nucleoside plus phosphate equals nucleotide. Got it.
And then these nucleotides link up through their phosphate groups to form the huge polymers that are nucleic acids.
Like DNA, the blueprint of life.
DNA, deoxyribonucleic acid. It uses the 2-deoxy-D-ribose sugar, a phosphate backbone linking the sugars together, and the four bases: cytosine C, guanine G, adenine A, and thymine T.
And it forms that famous double helix.
Right. Two polynucleotide strands coil around each other, held together by specific hydrogen bonds between complementary base pairs. C always pairs with G, and A always pairs with T. It's an incredibly elegant structure for storing all our genetic information.
Truly amazing. And RNA is its partner.
RNA, ribonucleic acid, is very similar but with key differences. It uses D-ribose sugar instead of deoxyribose. It uses the base uracil U instead of thymine, TU pairs with A. And RNA is usually single-stranded, though it can fold up into complex shapes.
And its job?
RNA has many roles, but a primary one is directing the synthesis of proteins. It acts like the messenger in the machinery that reads the DNA blueprint and builds the proteins and enzymes that actually run our cell.
DNA holds the plans, RNA builds the structures, both built on a carbohydrate foundation.
Fundamentally, yes.
Which brings us right back where we started. Neosporin and aminoglycoside antibiotics.
Exactly. That initial hook, neomycin, along with others like streptomycin and kanamycin, belong to this class called aminoglycosides. As the name suggests, they contain amino sugars, and they have glycosidic linkages connecting different parts of the molecule.
So, sugars with amines linked together. How do they work as antibiotics?
They are potent inhibitors of protein synthesis, specifically in bacteria. They bind to the bacterial ribosome, the cell's protein-making machinery, and cause it to misread the genetic code or stop synthesis altogether. This effectively shuts down the bacteria's ability to function and replicate.
Very effective.
Yeah.
But you hear so much about antibiotic resistance these days. Is that an issue here too?
Oh, absolutely. It's a huge and growing problem. Bacteria are incredibly resourceful. Many resistant strains have evolved enzymes that can chemically modify the aminoglycoside antibiotics. They might add acetyl groups or phosphate groups to the hydroxyls or amino groups.
They sabotage the drug?
Basically, yes. These modifications prevent the antibiotic from binding effectively to the ribosome, rendering it useless. A lot of current research is focused on designing new aminoglycoside structures that are less vulnerable to these bacterial enzymes, or finding ways to inhibit the enzymes themselves. It's a constant arms race. We should also mention erythromycin. It's another really important antibiotic, a macrolide, that also contains crucial carbohydrate rings in its structure.
A chemical arms race against bacteria based on modified sugars. That's incredible.
Yeah.
So, thinking back over everything we've covered, it's mind-boggling. We started with an antibiotic cream, and we've journeyed through energy, structure, genetics, digestion, disease, all circling back to these carbohydrate molecules.
It really underscores how central they are. What might seem like, just abstract organic chemistry concepts, aldoses, ketoses, anomers, glycosides, these are quite literally the molecules that build and power life. They're the building blocks of you, me, the food we eat, the world around us.
It's not just abstract chem, it's us.
It really is.
So the next time you say, eat a piece of fruit, or look at a sturdy tree, or maybe even, yes, put on some Neosporin, maybe take a moment. Consider that incredible, complex molecular dance of carbohydrates happening constantly, all around us and right inside us. It makes you wonder, what other everyday things are hiding this kind of complex, fascinating chemistry just beneath the surface, waiting to be explored?
Absolutely. We hope this deep dive has sparked some curiosity. Keep asking questions, keep exploring the world around you. There's always more to discover.
Thank you for joining us on this deep dive into the world of carbohydrates.